Oh, come on. Okay, good. It works. I just said how I know to operate with technology and I almost messed up. That's okay. So we are getting, so you just finished one of the probably most intensive quizzes in this first part of the class, like unit 1 to 5, because membranes are very hard. This is not easier, just so you know. But it's not as hard as unit 6. In unit 6, you all cry. That's literally the moment when people are getting depressed. But it gets better later on. Then you come to the nitrogen cycle, and then you want to shoot yourself. So again, I know I'm joking, so it's not like that really. It's actually very fun. At the end, you will realize how interesting all this is and how everything makes sense. So protein mobilization is a unit that will tell you a lot about how we get stuff out of the cell. And there are secretion systems and translocation systems, and we will make a clear line between that. But again, as everything in physiology, nothing is black and white. So things are interconnected. So that's something that I will keep repeating whenever we talk about some pathway. So how are prokaryotes mobilizing the protein? Proteins are most commonly exported from the cell, so this is what we will cover. And why are they important? Well, first of all, they're enzymes, toxins. Though them are proteins, they're dynamic and they're constantly moving in bacterial cell from cytoplasm to periplasm and usually from periplasm to throughout the cell. And that's very common for negative, gram-negative bacteria, especially because they have this very large periplasmic space. There are so many ways you transport proteins from cytoplasm throughout system. So again, first through periplasm and then to the outer system. And it can be secretion, which is related to secretion systems. And we will start with secretion system 1 today. And there is translocation. Protein translocation, two modes, co-translational and post-translational, will be very important and for your quiz and for your midterm. But secretion and translocation are not fully separated, especially because there are two secretion systems that are tightly connected to translocation. And those will be secretion system 2 and 5 that we will cover next class. So what will direct protein movement through cell is what the protein function will direct the location of the protein. So, for example, if protein is performing its function in periplasm, then the protein complex has to be transported from cytoplasm to periplasm. If a protein is performing the function out of the cell, then it has to go from cytoplasm to periplasm and then throughout the cell, for example, like some toxins, for example. So the whole point is that there must be cellular machinery that will drive protein to the proper location where it will perform its function. And as you can imagine, system exists in both types of bacteria, gram-positive and gram-negative. You can imagine because they all have different cell wall structures, transportation system will be different, and there will be some specific secretion systems that are gram-positive. But we'll talk about that next class. So it's like execution system 7, but just a spoiler. Anyway, so before we move into the like deep details about translocation, because all this class today will be on translocation, I do want to introduce a basic terminology, even though I think you already know this, but it will be a brief reminder. So translation and translocation, of course, not the same thing. So translation is the process that is happening on ribosomes when the synthesis of proteins during, like, it's a part of central dogma of molecular biology. Replication, translation, transcription, translation. So mRNA is translated into the proteins on cellular molecular machines which are ribosomes. So that would be translation. Translocation comes after translation, even though they're very similar in terms of pronunciation. So translocation means the cellular process that assumes driving a protein throughout the membrane from cytoplasm to periplasm or from periplasm to outside of the cell, depending on where the function is performed. And there are two basic, well, there are two translocation systems. And again, don't get confused. Secretion systems are something completely different than this one. So translocation systems that we are going to talk about in this class are second translocation systems. That has co-translational and post-translational version. That is easy. That one you will figure out very without any problem. But those two are main translocation systems in cell. When we talk about secretion system, there are 8 of them, but there is no secretion system 8. There is 1, 2, 3, 4, 5, 6, 7, and then so no 8. So but there are total 8 secretion systems, and they are very specifically organized cellular machineries, and you will see a lot of connections with transporters, which is the reason why we learn about membrane system before we talk about these systems. They are secreting proteins throughout the cell through the unique structure that is usually connected to the cell. The model organism that you are, oh, yeah. Oh, sorry, interrupt me, please. I get lost when I say. Second that. So they are translocation systems, and so what's your question? So you mean is it the forward? No, just for second. Which my God. Well, because also that section stands for twin arginine translocation sequence, but we'll come to that point. That's why it's called STAT. Okay, because it can double arginine as a sequence, but that will be covered later. All about that signal sequence that actually directs it, but it will be in a few minutes. So, and SEC you will memorize very easy because it's called SEC-JAG. So like our airport. So anyway, so the model organism E. coli, so pretty much a third of the proteins need to be moved throughout the membrane. So either they will be transported into the periplasmic or cytoplasmic space, but the reasons for moving these proteins, of course, can be very different. For example, if we talk about pathogenic E. coli, and there are things that are pathogenic for other pathogens, not only E. coli, that be moved throughout the system are toxic or hydrolytic enzymes, something that will attack host cells. There are some organisms, and if you took Bio 322 last year or you heard about it, there are geobacter systems proteins that are cytochromes because they make those nanovirus, so they are secreted with the purpose to respire solid metals. And geobacter does that, and that's used in microbial fuel cells. So that's another reason for secreting proteins, like respiration, energy generation. Very often, cells are secreting proteins that will enable attachment to other cells, like to form biofilms. And toxins are probably the most popular, especially in pathogenic microbiology, like cholera toxin, botulinum toxin, hemolysin. So these proteins perform different functions, like for example, hemolysin is inserting into the membrane for blood paralysis. Botulinum toxin is causing facial paralysis, but it can be actually used for other purposes. Think about that from biotechnology perspective. As a 38 years old woman, I'm thinking about Botox. I have to, like, I'm getting old. You're not getting, no. That's again where you employ bacteria. So anyway, and bacteria, so there are really, really different reasons why you would secrete proteins. Very often bacteria are going to attach to the cell surface thanks to these proteins. So, but everything actually starts from this peptide synthesis on ribosomes. And this is what the first picture tells. And again, I apologize if you already know all of this. I'm just reminding people who don't know. The proteins are actively translated on ribosomes. Like, again, I'm not going to go in details how our protein is translated, but I'm assuming you already know that everything starts from ATP codon here, peptide synthesis. And as protein is getting translated, it will be attached. So let's say you're translating a protein and it's going to be exported through SEC or TET system. So the way of interaction with that active ribosome will determine the type of the protein. So that, and we will see that in a few minutes. So again, some proteins can be exported to be integral membrane proteins, or they can go to periplasm to be hydrolytic, like a peptide degrading enzymes. But most of the time, if we talk, so there are proteins that fold in cytoplasm and get exported through cytoplasm in periplasm into the folded states. But there are other proteins that get translocated over the membrane in unfolded state. And there are different systems that will export these proteins. So if they are folded fully, so if they are translated on ribosome and then they are fully folded and exported through the cell membrane to the periplasm, they will be exported through that system. But if they are unfolded, then they are exported through sex system. So sex system, thus, in my opinion, is a little more hard to get. That is easy, very easy, but sec has two variations, and that's where a ribosome comes in. So, but bottom line, unfolded proteins exported through sec into the periplasm, fully folded proteins exported through TAC into the periplasm, and then later on to do their jobs outside of the cell or in the periplasm. So there are 4 steps in translocation. So again, just as a brief reminder, first step, of course, has to be translation in cytoplasm from ribosomes. So that's the first step. Protein has to be translated in cytoplasm. Next step is to sort that protein out. What that means is if it's going to be folded, then you will sort it toward that translocation system. If it's unfolded, you will sort it throughout the sec translocation system. Next one would be membrane targeting. What that means? So both sec and TET systems have some membrane components that will be different depending on the protein that is exported. So once you sort the protein, and usually you sort it with some signal, you will direct it toward the membrane. And the next one, of course, is an export, which goes, of course, to this very nicely designed membrane system. And very often you will have these chaperones that have very significant role in protein translocation, and we will cover each of them in a few minutes. So, SEC pathway component. So what is the SEC? SEC means SEC is going to export proteins that are unfolded. So completely different types of active-oriented proteins. So SEC pathway components will have these 3 proteins, and they look like integral membrane receptor that is called simple drag, like our international airport, SEC-Y, SEC-P, and SEC-G, or SEC-drag. So that is actually channel port. That's how it looks. And each of these 3 components, protein components, have specific role in the protein organization. So this SEC-drag pore is supported by a component called SEC-A, which is ATPase. It's a helper protein. It's not a part of integral membrane protein. It's inside of cytoplasm, but it mobilizes itself toward the SEC-drag, but it will assist into the protein translocation. You can conclude by name that there is ATP hydrolysis involved. So very often, in some specific cases, actually, ATP hydrolysis in SEC translocation is supported by proton motive force. So there are 2 types of energy used for translocation. And what's the structure? Again, SEC-drag is a channel pore, nothing special. You will see in the next picture, but it looks very cool. And each of these 3 components have specific roles. So intrinsic membrane pore is how the protein looks, and the Y component is actually the passage for protein. So I put channel in bracket because that's how it looks, but it cannot be like, I wouldn't like you to associate that with a simple mechanosensitive channel that we talked about. So it is because it does involve some energy. So Y components will be the passage for proteins. The one that surrounds a component is going to be E component. So like think about like coating the pore. So that's the second one in line. E. And the G is so if you think about the pore and then all around the little dots of sec E, and then what closes it is a G, which is the pore. And Jag is in the middle, but A, sec A is ATPase. It's a chapter. And you will see in a few minutes how that works. So again, you don't have to memorize how many transmembrane domains the sec-Jag has. It's just integral membrane receptor, and this is how it looks. It's embedded in membrane, so it's like a transporter. So just memorize what Y, what E, what G, and what A are doing, and memorize that A is actually part of the holoenzyme, but it's not embedded in membrane. So majority of E. coli proteins are using a translocated through sex system, like 96%. The other 4% of proteins in E. coli are exported through TAX system. Half of them, or 60%, actually remains in membrane, and the rest of it is going to be either periplasmic or will be exported like proteome. This membranome, it's a part of omics, like you know how we always talk about genomics, transcriptomics, metabolomics, proteomics. Membranome is a collection of all proteins that are in membrane, and I personally believe that this will be the most informative from physiological side point, because membrane is the master point. That's where everything is happening. So I'm personally very impressed with the cell membrane, and there's so many interesting proteins that you can find over there. Gram-positive, of course, have additional proteins, because if you think about how all that protein has to go through that thick cell wall, like peptidoglycan cell wall, obviously gram-positives have to have a specific transportation system. Very important molecules that participate in this translocation are chaperones. So why are they important for translocation? Because they're especially important for proteins that are not getting, that are exported unfolded. Because very often you will see proteins that are translocated in unfolded states through the cytoplasm, but you need to protect that unfolded protein from unwanted interactions. So once when it gets exported through the membrane into the periplasm, it actually has all chemically available bonds, so it can fall properly. So that's why chaperones are binding to it, and they actually protect that unfolded protein. So that's why they're so important. And this is literally what I just said. So that's the reason of chaperones. And again, that is easy. That is very easy, and I will briefly mention it in a few minutes. But where confusion usually happens, and where memorization comes to the point, is two ways of tech translocation. It can be co-translational and post-translation. And the name tells you that it is actually tightly related to the way it's translated on ribosomes. So either it's co-translational sec or post-translation. And let's go now through each of these each of these processes. Okay, so ready? Co-translational version. What means co-translation? So again, I will go in detail step by step. So as we know, protein gets translated on ribosomes. Everything is great, blah, 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 mRNA, rRNA, all gets nice. Now, during that translation, what will happen is that the protein will receive something called N-terminal signal sequence. As the name says, it's on the N-terminal end of the protein, but that happens during translation on ribosomes. And this N-terminal signal sequence is actually going to be specific markers for specific translocation. Now, stay tuned. Protein is translating, getting an N-terminal signal sequence, and still stay attached onto the ribosome. So ribosome is still there. Nothing is taken out from it, which is important because during this co-translational version, one part of the ribosome called SRP, or signal recognition particle, which is again part of the ribosome, will recognize this N-terminal signal sequence that was added during translation process. But protein is not completed yet. Some species have this so-called trigger factor that helps interaction between signal recognition particle on the ribosomes and proteins in translation. So it's like a interaction point, but not all of them have it. That's why I put it in the top. And now all this complex protein that has N-terminal signal sequence and is on ribosome and there is SRP involved. So all this now big complex during the translation, again, protein is not over yet, is going to be moved toward SEC-JAG complex that is going to sit where? On the membrane. SEC-JAG is in the membrane and waits for proteins to be translated to receive N-terminal signal sequence to be recognized by SRP on ribosome, and then all that like complex structure with ribosomes and protein attached travel to SEC-JAG. The protein that will wait on SEC-JAG is this dock protein FTSY. So it's literally like a docking protein on SEC-JAG that is waiting for the protein that will be exported. And this FTSY protein that sits on SEC-JAG is going to recognize this now upcoming complex. And based on this interaction to N-terminal signal sequence in this FTSY, you know that this protein is meant to go outside through SEC-JAG. How you export? So again, think about this. This is energy-intensive process. So this is one of these situations where you don't have ATP actually, but you have GTP molecule that will be hydrolyzed and you will release energy. And then this energy is going to be used to push the protein throughout the SEC-JAG channel. So again, SRP, we consider it chaperone that binds emerging N-terminal signal sequence. So, and all this SRP ribosome and pre-protein complex will dock on SEC-JAG channel. FTSY is membrane-associated protein, so it's not part of SEC-JAG, but it's close to it. It's like a docking protein that accepts all these complex SRP ribosome and terminal peptide. And there is no ATP hydrolysis; it's only GTP. And one thing that distinguishes this process from post-translation is that a ribosome remains associated until protein is fully translated and released. So, and SRP is the key landmark of this co-translation. So, all protein full with N-terminal signal sequence gets exported through the cytoplasmic membrane. And in periplasm, this N-terminal signal sequence gets taken out by peptidase in periplasm because N-terminal signal sequence is not part of mature protein. And the protein gets folded into the peptide. That's the key. Now, what about post-translational version? Again, the name tells you protein probably gets first translated, then taken by something and pushed to the membrane. That's the whole difference. So again, situation is very similar like for co-translational one. So protein is synthesized on ribosome, and again, it gets N-terminal signal sequence. So this N-terminal signal sequence will be associated to all proteins that get to be exported through signal, and also with cut, but cut has different entities. The key difference, once when protein is fully translated on ribosome, then the post-translational process, then translocation process begins. So you don't, in this case, you don't have all that movement of full ribosome and pre-peptide with 10 terminals to the membrane. It's done on ribosomes, it was taken out, has N terminals, and now it's ready to go. So this is where SEC-A comes into the game. So we don't have any more SRP, we have this SEC-A channel. So This second ATP hydrolysis, this molecule that I told you is a part of a whole enzyme, but it's not integral. It actually has a pocket for this N-terminal signal sequence that is on that translated protein. And this tag A literally touches it with that pocket, like recognizes that N-terminal signal sequence. So it's like analog to SRP in co-translational version. So this tag A now drags that protein to the membrane. And of course, in the membrane, who waits is sec-jag channel. Unlike in co-translational version, in post-translational version, you do have ATP hydrolysis. So it's not GTP anymore. Here is ATP. Because sec A is ATP hydrolysis, and ATP gets cleaved to ADP and PI by sec A. And again, unlike in co-translational version, you have proton motif force that is helping moving these proteins through the membrane. So in co-translational version, it's only GTP hydrolysis. Here, it's ATP and proton motifs. Again, ATP and proton motif force will push the protein through the membrane, and the first one that will come in contact with sec-jag will be the N-terminus, which again is going to help only to be exported throughout the membrane. Once when that protein is in periplasm, so N-terminal signal sequence will make the contact and will transport it into the periplasm. Then peptidase will cut out N-terminal signal sequence and protein will fully fold. So N-terminal signal sequence is important, but it's not part of your mature protein. It gets cleaved out in periplasm. And the N-terminal signal sequence has very specific features. It's a positively charged, which allows association with SRP and sec-A. Again, one in co-translational, the other one in post-translational version of sec export. And it is a substrate for peptidase. Located in periplasm will cleave out N-terminal signal sequence, which is not a part of mature protein. Again, it's only required for translocation, and it's there to mess up your open reading frame when you are cloning. Don't forget about that. That's just something that I really hate because it always messes up your open reading frame. So we don't like N-terminal signal sequence. We cut it out every time when we can. Again, just funny story. It does get coded. It starts with ATG. So God help me, I never knew how to do it. Yeah, I always mess up open reading frame, so I just cut it out. I can't do it. So one of the examples of N-terminal signal sequences are PHAZ enzymes, which are PHBT polymerases. They are used in degradation of biodegradable plastic. So they are produced with N-terminal signal sequence. Why? Because PHAZs can be exported to periplasm thanks to that N-terminal signal sequence because PHAZs have a lot of disulfide bonds, and disulfide bonds disulfide bonds are usually formed in periplasm, which means if you have a protein that is full of disulfide bonds, you have to put that N-terminal signal sequence because it has to be exported to periplasm so you can have proper folding of disulfide bonds. Unless you have specific type of cells, which is called Rosetta gummy, because they are modified genetically to form disulfide bonds into the cytoplasm, so you can cut out N-terminal signal sequence, and you don't have to bother with cloning it in the proper reading frame. And then your life is much easier, right? Never knows that. We were doing that recently, right? Every time you put N-terminal signal sequence, it's just a math. Everything is in math. Anyway, Archaea, as usual, I don't have much to tell you about Archaea. They have only co-translational version of protein of sex pathway, but majority of Archaea species are using TAT system. So, and I will talk about that in a few minutes. So, in E. coli, when you delete TAT system, that doesn't prevent normal functioning, but for Archaea, it needs to be present because Archaea are mostly using this TAT translocational system, and it The big difference is that this translocational system is actually moving only fully folded proteins. So what that means that it must be bigger. So channel must be bigger. And the process is pretty much similar. It's fully translated on ribosome. And during translation, you will again have this N-terminal signal sequence, but it will be specific type of N-terminal signal sequence, which is called twin arginine. And that's what it stands for, twin arginine translocation machinery. And this start sequence, again, twin arginine, allows association of the specific components of that channel. So that sequence is actually has the region that includes the signal site for cleaving off. So it's going to be cleaved off after it's interacting. So basically, the way how it goes, the protein gets folded to all mature structure. And then you will have this specific type of chaperone that will recognize TAT, which means twin arginine and terminal signal sequence. And that chaperone will bring it to the enzyme translocase in the membrane. So TAT translocase is in the membrane, and this TAT channel translocase is very large pore because proteins are fully folded, so they have to be exported. So channel have to be. That's all. So once when TAT is on the channel, this time there will be only protein multi-pores that will be used to push it through the pore. So again, a completely different sac. One of the examples that I just have briefly is halophilic archaea. So archaea that live in a very high salt environment. So most of more than 90% proteins are exported through that system. In E. coli, it's only 4%. So it's not really so important for E. coli. I mean, it's important, but more proteins are exported through this SAC system. So just a brief comparison of SAC and TAC. So again, bacteria and archaea have SAC, but TAC is mostly present in archaea. 4% in bacteria, don't memorize numbers, just know that it's a minority in bacteria. In TAC, chaperone-mediated protein that brings it to TAC translocase. In the SAC system, you will have SRP and SAC-A. So SAC-P is just analog to SRP, but you need to memorize only SRP. The key point is that SAC translocate proteins that are unfolded, while TAC translocate proteins that are fully folded. And for SAC, you will have ATP and GTP supported by PMF. So again, for co-translation will be GP, for post-translation GTP, for post-translation will be ATP plus PMF, but that system only uses PMF. And so first of all, I will stop here and ask if you have any questions.
Reminder that if you have questions after class, also feel free to post them on the question forum.
And Marisa will tell me because I will very often forget. I try to monitor it, but then I just fall asleep. I'm going to be completely honest. I just forget. But Marisa will remind me, and then I will respond, or she can, like she's equally good, so she will respond. Questions? Yes. Okay. What first?
Yeah.
Yeah. Okay.
Yes. Yeah. When does the bacteria decide to use the whole translational one?
Okay, let me just go to say.
Yeah.
So the question was, when does the bacteria decide to use co-translational versus co-translation? So it's not bacteria who decide, it's about the proteins. So proteins are genetically coded to be exported via one or another system. So there is no decision. Whatever the gene's genetic machinery says, if it has SRP associated N-terminal signal sequence, if the protein needs to, for example, there are certain proteins that will be exported in unfolded state. It really depends on the function of the protein. I could tell you exactly which proteins, but I have to search for it. Okay, Jacqueline. So from what I know, so Jacqueline's asked question in co-translation system that itself use PMF as well. I'm not aware of it, but it is not unlikely that there might be so. But from what I know, it's only GTP. More questions? Okay, so the question was about recombinant proteins that have to be folded in periplasm. So there are certain proteins that have a lot of cysteines, methionines, those amino acids that form, especially cysteines, because cysteines have that SH. So when S2 cysteines react, they give that disulfide bond. So SS bond is very tight, and very many proteins are full of these cysteines because they have very heavy structure. So the way how genetically is determined is that these sulfide bonds have a suitable environment to be formed only in periplasm, in regular E. coli. Okay, so if you have a protein, a recombinant protein that has a lot of these sulfide bonds, for example, superfolded protein GFP is one of them. So because it has so many disulfide bonds, if you don't put N-terminal signal sequence on that protein, it will not be exported to periplasm, which means it will not be properly folded because these sulfide bonds have to be formed in periplasm. But you don't put N-terminal signal sequence, so nothing gets formed properly. However, if you have specific type of cells that are genetically modified to fold those disulfide bonds into the cytoplasm, then you can skip N-terminal signal sequence because they will be properly folded because they are modified for that. They're just all cells genetically modified. Those type of cells are called Rosetta Gummy. They're most likely equal. They're very popular in recombinant protein construction because it facilitates energy. Like think about how much energy you have to invest to transport protein across the membrane versus just put it inside the cytoplasm, gets translated quality all good. Does that answer the question? Good. All good questions. Okay, so I am a nice person, so I will let you go because we have 8 minutes. Oh, yeah, we have 8 minutes. You can.
If anybody wants to see it, do you want me to review the quizzes? What's the consent? Yeah? Yeah.
Okay, then well, you can try now starting, but I would let's do this because next lesson is not going to be hard. So you at the beginning of Thursday we review quizzes again, so you can go nicely over it. Is that okay?
We can get it out.
Like you have only 8 minutes. Yeah. So on Thursday you again have Marita first 15 minutes, and then we will finish with protein secretion system. It's not hard lectures, so it will be easy. This one is much harder. Okay. So you have again Marisa next Thursday. That means I can. No, no.